So hello and welcome to the Shiki Science Show, where today I'm joined by Dr. Timofey Glinin, a postdoc at UCSF who's been involved in the longevity space from a unique angle, trying to make research and data sharing more open. We discuss his contributions and how his new venture, First Approval, is building the infrastructure to overcome these additional barriers to longevity breakthroughs. First Approval is also now hosting a competition to incentivize and accelerate not just longevity research, but all of science. I hope you enjoy our conversation. So we're both researchers and also have a lot of interest in the longevity space. And of course, there are many challenges of being a researcher, and of course, biology itself is very complex. But what do you think are some of the maybe other major barriers in biological research that perhaps people haven't thought about?
Yeah, I really interested in biology of aging for more than 10 years, and we explored different methods of understanding this phenomenon. So we wrote review articles, we wrote books, we invited really relevant scientists on conferences, interviewed them. So we tried different approaches, and we created a database, open gene database, which specifically focus on genes, how they influence on aging process. But we still don't have all answers about aging, and we still, for example, could not find the priority of drivers of aging. So we know that some mechanisms involved in this process, but we still couldn't prioritize which is more prominent and which is less. So for all these ideas, we try to think and find additional bottlenecks in aging field. And one of bottlenecks that was illustrated, for example, by LBF survey, longevity bottleneck survey, is that for scientists, the unavailability of publicly available data sets is one of the biggest struggle in the field, and they put the priority for opening data about aging, opening data sets about aging, even higher than regulatory changes and increased funding. So we understand that potentially data availability could be the problem that we can solve much more easily compared to, for example, increasing funding or other big experimental projects. We can just share existing data in a much more detailed way with great annotation and somehow involve scientists into this open data movement. So it could be some solution that we think about and which is kind of new for the field.
Yeah, I think that's a really important point. You describe it as open science, and perhaps I'm naive in thinking that open science is purely about open data sets. Maybe you can correct me later on that, but is it maybe for us it's obvious that sharing data sets and having availability to it is important, but perhaps to my audience, why is it so important that we can access data sets?
Yeah. You're absolutely right about difference about open science in general and open data as particular field, and I'll try to describe what I know about it. So open, science, basic of the science is reproducibility of results, and we know that today biology have not as high reproducibility as we want to have. Some studies show dangerous numbers that about only 30% of scientific articles could be completely reproduced.
And that's too high. It's too high.
Yes, some illustrate a more mild picture, about 50% reproducibility rate, but still these numbers is really harmful, because this means that a lot of additional research could be done over the article, which was initially like not so clear, and this means that these researchers could also struggle about some problems inside like hypothesis of these experiments. So it could be like really complicated thing to understand what is the reason of this reproducibility problem. But definitely the first step would be to look at the original data, because in some hand, article is interpretation of data that scientists get. So they generate some experiments, they found some results, and then they analyze these results, try to combine different experiments in the single story, and during this process they somehow interpret what they get, and only this interpretation is available in scientific article. But if you get the data with detailed description what was happened, other scientists could explore this on their own way, which could give them additional insights in like unexpected areas, because they could be interested in some, for example, you show data set, you show like big number of photos of some samples, and for you as primary researcher, the goal was like to determine some protein, for example, or anything else in tissue piece. But for other scientists, they could re-explore your data based on, for example, membrane structure or anything that they could see interesting in your model that you used. And from that perspective, opening of the data is not only answering the question what problems in case of reproducibility, but it is additional opportunities for get insights from independent reuse of this data. And additionally, it gives you opportunity to merge data sets and get larger like N for your experiments, and you can improve methodology by getting additional data together. So from that perspective, it is like relevance why data is important. Additionally, for aging field, I can mention that lack of paradigms, of single paradigm in aging, and a lot of hypotheses give us the situation where different scientists work on different concepts, and they like commonly don't take too much attention to other theories of aging. For example, if I perform experiments in the area of epigenetics of aging, I could be focused on this particular like results and experiments. But if the person who are interested in, for example, extracellular matrix will explore my data in case of his own concepts, he could find completely different interpretation of my result. And it is the situation where lack of general theory result in even much more importance, much higher importance of disclosure of data.
Yeah, I understand. And so if my understanding is correct, you're saying that if I have a data set, how I interpret that data set could be very different to many other people. But in that case, do you think that our interpretation is the reason why there isn't this reproducibility, or like what is the source of this lack of reproducibility in science?
it could be a lot of reasons, and open science try to understand all these reasons. And like data availability could be not the reason why experiments are not reproduced, but without this data it could be much more difficult to understand what's happened in reality. So like if you will start think about what was the problem, the first thing that you need is data, and it will help you a lot. But speaking about reason why experiments are not reproduced, sometimes it could be some misinterpretation of statistics. So you can use like incorrect statistical method, or your colleague who tried to interpret your result could also make a mistake in statistical interpretation. So it could be both-sided, and the good strategy to solve such kind of problem would be to look at the data that is generated by both researchers and compare them and understand how like second data set will look like applying different statistical method. Other reason of problem with reproducibility could be so it could be different model organisms. So you use one strain of mice and it is not reproduced on other strain of mice. You can use different reagents, so it could like be really crucial for reproducibility. Sometimes it could be methodological problems. So you just get something that like has artifacts. Looking at the data also could tell independent researcher like who made these artifacts, for example, do it in original paper, or it is something that team who tried to reproduce made like incorrect. So all these potential reasons, and probably it is like I described at least more than 50% of all cases that is not reproduced. In all these situations, get access to original data is crucial, but the problem that scientists sometimes could even forget what was in original data, because if they don't organize this data after the experiment, or at least before the publication of original article, these files could be lost, could be like the persons who run this experiment could leave the laboratory. It could be a lot of just like time went away and you couldn't remember one abbreviation on your data, and you are not sure about what's happened then. And because of this, like requests of the data after the article publication is often really big challenge for both data users and data authors. And from that perspective, the culture of like good practice, where you publish data after experiment, or at least during the article, is much more valuable, because you can avoid a lot of mistakes with data annotation from that strategy. And journals allow this, so they you can upload data in repository primary to original article publication, and every article in good journal have specific section which called data availability section, and you can just integrate your data set inside this section. So It is something that publisher also think about and try to improve, because if the data is available, it regularly means the increased attention to this publication, increased citation number, and this will drive the journal to get a high impact in scientific community. So
yeah, exactly. And so I mean, we've mentioned the word dataset quite a few times, but I feel like we should perhaps try and explain what type of datasets or different types of datasets biologists might have, and perhaps explain on the difference between a raw dataset and like metadata and the sort of final published datasets.
So it could be different types of classification, what dataset is and what we can interpret and classify it. But for me, the dataset is data outcome from every scientific experiment. So if you run small experiment, the outcome could be just single photo of your gel, for example. If it is different type of experiment that generate like big data, the data set could be terabytes of information, and it could be, for example, transcriptomic data, single cell sequencing, RNA-seq data. It could be genomic data. Sometimes it could be microscoping experiments where you like screen several set of slides, and you will create the graph and statistic based on these illustrations. And in that case, this set of photos that you will use for your experiment will be data set general from your experiment. So from that perspective, we definitely could classify the data sets as big data and like small data sets, and but both are relevant, because sometimes even single photo, raw photo from like Western blot, for example, could be result of big problems with which will be problem for numerous of laboratories which wouldn't know how to interpret it. So we have these examples in like real life. And speaking about dataset, it could contain raw data, which is primary files that you get specifically from device which you used during your experiment. Or it could be processed data, which is something that you worked on. For example, you export like TIFF files from proprietary microscope file, or you perform some filtering or sorting the information, and or even some kind of initial analysis of this data. All this will be processed data. And dataset could be only raw data, could be only processed data, and could be mixed, where you put all raw and processed data. From our perspective, it is much better when both raw and processed data are available, because it will give additional insights about your methodology, about how you worked on your results. And this is like the best if you include all, but at least any kind of data set is much better than absent of this data set. So from that perspective, even, for example, raw data with good annotation is much better than nothing. The last thing which I will say, that data set definitely contain data, but it also contain annotation, which could be annotation of different levels. It could be annotation of exact files. For example, this photo was captured and you put the date, you put the device, you put the information about magnification of, you can put maybe information about microscope, about laser intensity. You can make a lot of efforts to annotate the exact file, and it is what regularly called metadata also, metadata of files. And simultaneously you can put annotation about, for example, design of your experiment. So you can propose that it was 2 groups, it was samples that we received from, for example, mouse specimen or from human cells cultures. You will describe how treatments was processed. You can describe what was the initial idea for that experiment, what you was expected to test, and finally it could be some kind of interpretation. So for example, sometimes scientists could already know that the data is negative, and they could indicate why this data was negative. For example, everything in methodological part was correct, but they could make some notes, and they could make, for example, interpretation of whole experiment, or they can specifically mark information that this particular sample was, for example, processed faster than all the others, for example, and we decided to include it in our data set, but it is possible that other scientists who will explore this data will find that faster processing time could result in something specific, which will give insights to them, but doesn't necessarily mean that the initial data interpretation was problematic. So
yeah, exactly. So I think you raised actually quite a few points there. So firstly, about experimental design, also I know how important that is, and of course you can either have, I think all good scientific data needs to have the right experimental design, but of course you then also say bad data or good data, the annotation is just as important, because without the annotation it's very difficult to reproduce it. And so I think about these problems myself all the time, and you mentioned how we need to annotate all this information, the magnification, the intensity of the light, the exact reagents, the concentrations. That's a lot of manual stuff for us to do as researchers, in addition to the actual scientific thinking and actual hands-on experimental research. It's quite a massive task and quite a big ask for researchers to do. Now, of course, good researchers should do this, but is there solutions to help facilitate this process? And so nowadays, I'm thinking, how can I use AI tools to help me, check my experimental design, or also encourage me to be better at data annotating as I am actually doing my experiments. But what other solutions do we have to solving this massive problem?
Yeah, it is a really great question, because one of the main reasons why scientists do not publish and share data is because of efforts that they need to put for this process. And one potential solution could be that the annotation is much more easily if you perform it right after the experiment. In any case, you will need to write some kind of report. You need to record for yourself what was done for future publication. So you in any case will somehow do at least part of this work. And the good thing about making these data annotations like regularly after the experiment, that you can use, so after you make one annotation of experiment, you will have template that you can use for other similar experiments, and you will need only focus on difference from initial design to new design, and it will somehow could standardize your work for yourself, for example, because if you have these detailed protocols, what was done before, you somehow clear your mind and open for new experiments, because you don't think that, yeah, I'll get to this later, but I need to remember some details. And it is high probability that if you will return to some experiment, for example, half a year after experiment was done, and when you just write the article about it, you will feel a lot of problems with something that you remember that you write it in some lab notebook, but you need to find where it is, or you expect that you will find this information. So it is something that scientists should take into account, that if this practice will done immediately, it could be much more simple, and it could simplify the life in the future when you will need to publish it. So it could be one solution that didn't require any additional toolkits. It's just something practical. I think that AI could be used as helpful tool to understand what information should be put into data annotation, because experiments are pretty standard, and regularly AI really perfectly know which details are essential for both reproducibility and just correct description of every detail of experiment. And this tool could be used. Additionally, some projects try to create community standards for data annotation, and I can mention, for example, CIDAR initiative in Stanford, which try to create complete standards for different laboratory experiments. So the idea that this number of characteristics that you're able to put in your experiment is already set it up for you, and you just go through this list and understand what additionally you should put about your experiment to text of annotation. Some online lab notebooks would be useful tool. So if you make a good and ongoing work of writing what's happening in your lab, it could be really useful tool also. And definitely it is great solution if we can find some more automated way in one hand and more beneficial way to share data. And I think the problem of efforts about data annotation is some kind equal to less of benefits from data annotation, and because if, for example, you get some grants based on how accurately you annotate and share data, it could be great motivation for scientists if they understand that their activity could be somehow transformed into money, for example, into future grants, into like recognition of scientists, into like a greater position in future career, it will be great motivator to like make data annotation much more comprehensive and detailed. And like this ideology that we can not like force scientists to this ideology that we can not force scientists to just spend a lot of time for data annotation, but just increase the rewards that they get from data publication, could be like one of the solutions how we can transform this situation. And specifically, a team of first approval project, which I am a member of, is focused on testing such kind of hypothesis about potential benefits that we can bring for scientists to make the data annotation and publishing process much more interesting for them. One possible solution could be more like curation requirement. Scientists want to know what's happened with their data after they publish it. They want to, for example, get more impact from their data publication. They want to guide the users who will download their data, and they want to check the correctness of analysis of this data. So it is what we hear from scientists from everywhere. We have questions about future of my data set, so I'm not hurry up to publish it. I want to know how other scientists would like to analyze it, and only if I'm sure that I know and could avoid mistakes from this scientist, I can share data with them. And potential solution could be create the infrastructure where data is available, but data author guide the reuse of data, test the correctness of data annotation, and participate in future articles that interpret this data as a co-author. So it, like from our perspective, could be really powerful solution, because scientific articles is really highly valuable, high value for every scientist. And collaboration itself based on your data set also great benefit, because you don't know which scientist will be interested in your data. It could be scientists that you can, for example, invite to your lab because it's brilliant young scientist, or otherwise it could be really great and well-established scientist who somehow interested in your data, and it could be great collaboration, and you can propose additional experiments together. So collaboration is something that also could be really great driver for data disclosure. So, but it requires a lot of efforts, and potential more simple way of make benefits for scientific community by data sharing is just to create direct, like choose the auditory, for example, students which would like to get award for career growth, or would like to get publication for demonstrating how they know the methods, for example, or just students all the time want to have more publications. And if you will create the system where they would not just publish the data, but they will participate in some kind of contest where they could get an award or diploma of excellence that will nominate the excellence of their dataset. It could be a solution which we try to bring to this world.
Yeah, exactly. And so you mentioned quite a few things now, but I believe through first approval, you're trying to address some of these issues. And as you mentioned, this competition, you're currently running a competition aiming more for students to try and help price them based on not just the outcome of their biological data, but actually on the data set itself, how it's annotated. So perhaps could you explain first sort of the origin of first approval and how that came to be?
Yeah, origin of first approval is so the community of scientists who are interested in acceleration of research in biomedicine, first of all, but in general science also. They unite to create a repository and data publishing platform which could specifically focus on increase benefits for data publication. And existing things that if you publish data on first approval, you will get PDF publication with DOI, which you can share with anyone, and this publication is generated fast and automatically, so you can publish data, you can get this PDF file, and additionally you will know who are interested in your data, because it will be indicated which members of community, which users download your data set. So it is what this platform is already existing, and you can use it at firstapproval.io, but the team is working on bring more direct benefits for the community, and initial project is student biological data competition, which focus on publishing data sets from undergraduate, graduate, and PhD students. It could be either processed or raw data or merged together. Every participant who perform like data submission and after editorial check will get data publication with DOI. So it is like direct outcome. If you submit your data set, you will get publication. The second is that you can win the award, and we have money prize fund of 7.5 thousand USDT, which we will spread by 3 tracks: undergraduate, graduate, and PhD students. Each best dataset in their category will get 1.5K of dollars, and in each track, the 10 persons will be awarded with money prize. Additionally, we have 2 special prize, one for negative dataset and another one for replicative dataset, which is during submission, you can specifically indicate that your data set is replication of other experiment, or your data set is negative because, and you will describe why it's not support the initial hypothesis of experiment. So you can win both, like for example, a PhD student award and best negative data set. It is also possible.
I see. And so the competition is purely based on the data set itself and perhaps the annotation. There's no, like, will there also be interpretation of the results, or purely just how the data set is curated?
We look at the design of experiment, the background and initial hypothesis which was put into this experiment. So it is one of criteria to establish the best data set. The second criteria is the quality of annotation, how detailed it is, how how reusable and it is, and how much details you put to describe data accurately. The third criteria is the files itself, how easy to understand how your files related to your annotation, and if you put like raw and processed files, it would be better than if you put only raw files, for example. And so we check the files, and it is third criteria. And the fourth criteria is potential for reuse of dataset, which means that we just look, and if we believe that this dataset could be the great source of inspiration of other scientists, we will get more points by this criteria. So these four criteria, doesn't matter what exactly mean that you found in this experiment, and the challenge was to evaluate data, because it is not standard procedure in scientific world, and we create a great executive panel of judges. It is 5 professors, 3 from top United States universities, including Albert Einstein College of Medicine in New York, and we have one university in the United Kingdom, one professor from university in the United Kingdom, and one professor from City University of Hong Kong, from China. So we would like to make this competition open for students all over the world, and we encourage them to submit data sets. We have great organizing committee who will help to evaluate submissions and perform initial sorting of and organizing all submissions. And we have a lot of volunteers, and if you would like to be volunteer and just spread the word, for example, inside your university, also you can contact with us, and we really happy that a lot of people support our initiative. Some universities already shared information on their websites. For example, Cambridge University already invited all their students to participate. So
Yes, I mean it really is quite a unique and important competition. I mean, just besides competition, just what you're trying to establish. But it made me think that, do you see a future perhaps in 5 years, 10 years time, where instead of the journal papers itself that have this sort of more story narrative perspective to them, instead we'll see that raw datasets or datasets perhaps published on your site instead have more citations than the sort of more polished sort of story-like papers?
Yeah, I think that, so everyone in open science know that there are a lot of problems with scientific articles, and these problems arise just because it is like the main thing of current scientific process. Nobody really cares about how much experiments you have done which was not integrated into your scientific articles. Like journal publication is the main way of sharing with community what was done, and funding decisions based on journal articles primary. Like everything is based on journal articles, and this makes the situation a little bit difficult, because if the only one criteria in the system drive all the understanding, so I will say in other words. So if the journal articles is the only way how scientists could get attention to their results and get grant funding and get a position in a laboratory, so this drives the scientists to focus only on papers and not focus on other parts of scientific process. And we believe that this new standard, so we just would like to create additional system of reporting the progress, which not eliminate scientific articles, because you already and in future can just put information about your data set into your article. It's not compete with each other. But if you will create the infrastructure of data, and for example infrastructure on collaboration based on this data, it could drive the science in the way where working on data set and publishing of data set could be also valuable. So you can focus on data publishing, and it will give you some benefits, and or you can focus on work with already available data, and this will drive your own research, and simultaneously you can publish the articles in context when you ready to perform your invention in the single story with a lot of details. But the future could be that you, even in your article, you can use a lot of data sets from other researchers in this collaborative way.
Exactly, yeah, that makes sense. They're definitely complementary approaches. As you say, I could have my research article that has my data set, but also I can then compare it to other published data sets and come up with, again, my perspective. And I think personally as a scientist, despite as we said at the start, the interpretation of the data can always be slightly different depending on your interest. And I think in my case, sometimes that interpretation or perspective is the most interesting thing in scientific research. So I completely see how they're complementary. It sounds like first approval is very important, what you're trying to do. Also, of course, very challenging. And so how do you see it going in the next few years? What is the big plan for the first approval?
We would like to make introduce to scientific community a lot of different benefits, and we would like to communicate with different scientists and find specifically what they are interested in to make their data available. And I think that in future years we will try to improve and scale these methods. For example, now we run only biological data competition. I hope that this initiative would be annually, and we additionally could make the same open science solution for chemistry, physics, ecology, and like a lot of other areas where data is important for research. Additionally, we will introduce this collaboration system where scientists could publish data with collaboration and authorship requirement, which means that we will help scientists to collaborate with each other based on data sharing. Additionally, we have ideas to perform some kind of specificity in case of different data types, and we can expand from single first approval platform to set of curated specific repositories which focus, for example, on only some types of data or some specific areas of experiments. As testing project, we are preparing to launch agent data collection, because there we can accumulate specific data only from agent research, and it will help scientists to communicate and see and find valuable data specifically for their own field. But we are open to any other ideas and initiatives, so we would like to scale this initiative also. And I hope that in future we will have a lot of communities of scientists around first approval who will work together on the topics they are interested in, and they believe they could improve by their efforts in case of open science and open data.
Excellent. Yeah, I think I can only wish you guys the best of luck. And of course, I think not only is it important and specifically important for longevity research, but just in research in general. So I'll put the links to the website and the competition in the description below. But it was just great to speak with you today.
Thank you, everyone.